your PowerPoint presentation, share, and your answered questions. So everybody has to do all the same thing where they have to answer all the questions. If you're not presenting, you have to create your own to participate in the discussion. And if you're presenting, then you also present, right? And everybody's got to take a turn in the group actually presenting. Yeah, okay. Cool beans. All right. All right. So make sure you check in. All right. So how did the vampire know he was sick? He was coughing. He was coughing. Yeah. Not as good as last time, but you know. All right, let's see. So we're gonna start by finishing photosynthesis, right? Make sure you check in on iClicker. Make sure you do that. It's just attendance. You don't need the paid version, just the free version. Okay. So what we talked about last time with photosynthesis were basically the light reactions, right? The ones that are dependent upon photons coming in and stimulating those chlorophyll molecules and all that kind of stuff, right? That was the light dependent. Now we're going to talk about the dark reactions. But what do we mean when we say dark reactions? Light independent. Now, are you going to do these reactions in the day or in the night? HT, light. You're going to do these in the day. You're really not going to do these at night because you won't have the precursors that are needed in order to make this go, like ATP and NADPH, right? You make ATP and NADPH in the light dependent reactions. You're going to use them in the dark Calvin cycle. Well, if it's nighttime and you're not making ATP and you're not making NADPH, can you run the Calvin-Benson cycle? No, you can't, right? Okay, so what does the Calvin-Benson cycle need to run? Well, we said it needs ATP, it needs NADPH. What else does it need? It needs CO2, right? We need a carbon to make sugars, right? Sugars are carbon-based molecules, right? So we need those carbons in CO2. And then we're going to use them in order to make some 3-carbon sugars. From those 3-carbon sugars, we're going to make some 6-carbon sugars. But here's what's kind of interesting. You know, the Calvin cycle is a cycle like the citrate cycle. So what was one of those first substrates going into the citrate cycle that was a product from before? Do you remember? Well, acetyl-CoA comes in, but what was a product of the cycle that comes back and acts as a first reagent? Y'all were saying it. Oxaloacetate. So in that similar way, we're going to have a pentose sugar that is going to come in and act as a substrate in the very beginning of the Calvin-Benson cycle. So you actually need to have a sugar there to make more sugars. You can't start from nothing, okay? All right, so once you go through the Calvin-Benson cycle, I kind of want to give you all an overview of kind of what's going to happen before we go into the details of it, right? So what could you make sugars for? What's the point? Well, these hexose sugars can be used for 3 different things. We can use it to make sucrose. Sucrose is there for energy metabolism, right? Like the plant cell has immediate needs for energy, and so we convert it to sucrose. The other thing that can happen is there's enough energy, the energy charge of the plant cell is very high. So instead of using it for energy, we're going to store it as starch, right? Because it's going to get dark. We need to have some storage for the dark. The other thing is it can be used to form cellulose. Cellulose is part of your cell wall in plants. So those are three big uses of those hexose phosphates. Now, those hexose phosphates can be turned into pentose sugars, right? Those pentoses can be used in one of two things, either to go and build DNA and RNA, or re-enter the Calvin-Benson cycle. So you kind of have to know where all the different things can go. Good so far? All right, I am going to skip ahead to the end of the section, and then we're going to come back to this because I actually want to give you guys an overview. Here we go. An overview before we really talk about all the details. So when you look at the Calvin-Benson cycle, it's really what I call carbon shuffling. You're just shuffling around carbons. So if you can kind of count the carbons and keep track of the carbons, you'll understand what's going on. So the substrate that's going to be a product of the Calvin-Benson cycle that is also one of those initial substrates is called ribulose 1,5-bisphosphate. So if you look at this molecule, how many carbons are in ribulose 1,5-bisphosphate? Count them. 1, 2, 3, 4, 5. So this is a 5-carbon, so go C5, right? Now, in order to make a complete cycle, you're going to need 3 of them. Okay. Now, in order to run the Calvin-Benson cycle, we're going to try and make really 6-carbon sugars, and then we're going to break those 6-carbon sugars into 3-carbon sugars. So we have a 5-carbon. We're going to have carbon dioxide come in. Because I have 3 5-carbon sugars, I need 3 CO2 molecules, right? So I'm going to have 3 C1 molecules. That's my carbon dioxide. Are with me so far on counting? Okay. All right. Now, what we're going to do is we are going to convert it in what's called the stage 1, the fixation stage. We're going to convert it into 3 6-carbon sugars. But we don't make 6-carbon sugars in Calvin-Benson. How many carbons are in those chains at the end of the Calvin cycle? 3 carbon, right? So we're going to take these 6-carbon molecules and we're going to break them. So we're going to end up with 3 carbon molecules, but how many are we going to end up with? 6 of them, right? So those 3 carbon molecules are going to undergo stage 2, which is reduction. We're going to phosphorylate them. We're going to reduce them with that NADPH, and we are going to end up with, again, 6 3-carbon molecules. They've just been phosphorylated and reduced. Okay, so it's just changing the the biochemical properties of those 3-carbon chains. Now, what's going to come out of here? Look at this. This is kind of amazing to me. From this, only 1 of those 3-carbon molecules is going to be used to go on and make all those things we talked about. Only 1 of them. So other 5, so we have 5 3-carbon molecules that are actually going to have to be converted back in the regeneration stage to remake ribulose 1,5-bisphosphate. Because how do I go from 5 3-carbon molecules? All I'm going to do is rearrange using ATP. This is the ATP dependent process. And I'm going to make 3 5-carbon molecules. Do you see why I call this the carbon shuffle? So it's really 3 phases. The first phase is our fixation, where we incorporate that carbon dioxide into our chain. And then we cut the chain in half, right? Then it undergoes reduction, where we phosphorylate it, we reduce it. Only 1 molecule gets shipped out for use, and then it gets regenerated. Now, the delta G for this is very negative. It will happen, but it happens extremely slowly, very slowly. So how do you get around that? I mean, this is enzyme driven. You can't speed this up any more than it is. What did I tell you about the primary enzyme that does this process? Remember what I called it? Rubisco? B-I-S-C-O, Rubisco? It was the what? Most abundant protein on the entire planet. That is how we get around this being slow. Even though it has a negative delta G value, it's slow. So we put a crap ton of Rubisco in the plant cell, and we can have enough of a product that this is a useful mechanism. Isn't that really cool?
Just don't show those 6 carbon.
I'm gonna show you, like, we're gonna go through all the detail, but I really wanted you to know where we were going before we get to the nitty gritty. But yeah, in this diagram, between here and here, they don't show the 6 carbon, but I'll show it to you in just a second. Yeah, I really think they should, but it's okay. It makes sense if you show it. All right, now I'm gonna go back. Y'all ready to go back? See all these reactions, right? Okay. So even though we call it the dark cycle, it is controlled by the amount of light the plant gets. If the plant's getting light, Calvin cycle will go. Plant's not getting light, no Calvin cycle. So the products of the Calvin cycle, we said were 3-carbon sugars, right? You have 3 different 3-carbon sugars that are going to be generated from the Calvin cycle. 3-phosphoglycerate, glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. Each of these 3 can be used to make 6-carbon sugars. So C6. Fructose 1,6-bisphosphate and fructose 6-phosphate. All right. So remember how we looked at that wheel and we said it was really 3 major steps that occur, right? The first one is the synthesis of the carbon 6 molecule, right? This is one that they didn't draw on that wheel. And the Rubisco enzyme is the one that catalyzes this. And so then we're going to cleave that 6-carbon molecule into 2 molecules of 3-phosphoglycerate. 3-phosphoglycerate is a C3 molecule, right? Then what was the second stage? We had fixation. So 1 was fixation. What was next on the wheel? Reduction. And what was the last one?
Regeneration.
Okay. So in reduction, we have our 3-phosphoglycerate, and it's reduced to glyceraldehyde 3-phosphate. And this is the step that we need the ATP and the NADPH from the light reactions. And then at the end, it really doesn't say it, but one of those 3-carbon molecules is kicked out, and the rest are used to regenerate ribulose 5-phosphate. Okay, so that's the carbon shuffle. So here, right, we're going to make glyceraldehyde 3-phosphate. So what do? How many of those do you make? Do you remember? 6 3 carbon, right? So from these 6, 5 are going to be used to regenerate ribulose 5-phosphate. 1 is going to go on to do all those different things that we talked about, right? We'll say metabolism.
But that's the starch and the cellulose.
Right, right. Cellulose, starch, what else? Sucrose. Yeah. And then, yeah. Okay. So here's our overall net reaction, right? So when we talk about this, we're always going to talk about it. Remember how we did the citrate cycle and we had to count and make sure that we had enough of everything to make the cycle go. So sometimes you had to have 2 different things, that kind of thing. That's what we do here, right? So we said in the cycle, we're going to incorporate 3 carbon dioxide molecules. Those 3 carbon dioxide molecules are going to react with our ribulose 5-phosphate, right? So if you look, RUBP is the same thing. They abbreviate it because like writing it so many times is going to be a pain in the butt. But this is our ribulose 5-phosphate, RUBP. So it's not Rubisco, it's not the enzyme. RUBP is not Rubisco. RUBP is ribulose 5-bisphosphate. It's actually a bisphosphate. Ribulose 1,5. It's actually the bis form. That's where the B comes from. Okay. So then we're going to need our NADPH from the light cycles. We need our ATP from the light cycles, and we need water. And we're going to generate glyceraldehyde 3-phosphate. That's what our product here is. 3 of these ribulose 1,5-bisphosphate molecules. Then we're going to make that reduced form, right? We go from NADPH to NADP+, and then our ATP goes to ADP and PI. Right? All right, so let's look at these reactions kind of in a little bit of detail. Now, do you have to memorize all these different steps? No, please don't. Please don't. Please understand the general process, the big intermediates, but all of this is not necessary. So stage 1 fixation is done by Rubisco. RUBIS, Rubisco. Can't spell, I really can't spell. Rubisco. This is the enzyme, Rubisco enzyme. Okay. So this Rubisco enzyme, this is where you're really looking at the rate limiting step, right? And this is a very negative delta G value, about -35 kilojoules per mole. But it's slow. So like of all the total protein in a plant, like if you took an entire plant and you took the mass of all of the proteins in the plant, about half would be Rubisco. Okay, so you're going to start out here with your 5-carbon Rubisco 1,5-bisphosphate, right? So it's been phosphorylated already. That was an ATP investment, right, to regenerate it, if you kind of remember that from the end. We're going to add our carbon dioxide here, right? So we're going to have that. That's our fixation, really. So we're going to carboxylate our double bond here, right? And then we're going to go through a hydration step. So this is why we call it basically, you're really adding 2 molecules. You're really adding carbon dioxide and you're really adding water. So remember how we said that those 2 things were important in the net reaction? That's where these 2 come in. So then we undergo our hydration. Now we have how many carbons in our chain, right? After we added our carbon dioxide, we have 2 intermediates that are how many carbons? 6. That red one is the one from carbon dioxide. So now we have 6 carbons inside of our molecule. It's not a straight chain, but we still have 6 carbons in there. So now what we're going to do is we're going to undergo an aldol cleavage, and that event is going to give us 2 3-carbon molecules. Now they're not exactly the same, right? But this carbanion here undergoes protonation, and now we have two molecules that look exactly the same of 3-phosphoglycerate for every one initial ribulose 1,5-bisphosphate. But we didn't really start with one. How many did we start with? 3. So how many do we end with here? Really 6. It's kind of important to keep track. It's kind of difficult, but it's kind of important. Okay. So now we have to go through reduction. So now we've just got our 3 carbon molecules. This is going to take an investment not only of ATP in order to phosphorylate our 3-phosphoglycerate, but also we need our NADPH here to undergo a dehydrogenase reaction. So you're going to have both of those investments in the reduction phase. So we're going to go from 3-phosphoglycerate to glyceraldehyde 3-phosphate, which can then go through an isomerization reaction to make our dihydroxyacetone phosphate. Right? So simple isomerization. So what kind of carbon molecules are we throughout reduction? 3. All we're changing really is phosphorylation and reduction status, right? Okay, so, oh, that got smaller, huh? All right, so we've been through Calvin-Benson cycle, right? We've made glyceraldehyde 3-phosphate. We've made dihydroxyacetone phosphate. What are you gonna do with it? You could use an aldolase to combine the two and make 6 carbon sugars like fructose, glucose, and turn that into starch for energy storage. That would be a good thing to do, right? For when it gets dark. Now, the other thing is maybe your cell needs energy right now. So what you do is you use this PI triose phosphate transporter. So you know you're going to need inorganic phosphate for those phosphorylation events. So it's an antiporter. When one of those inorganic phosphates enter the stroma, then you're going to have one of these 3 carbon sugars is going to be transported out. Now that we have them out, we can either use them in glycolysis, right? Or we can turn them into different things depending on what we want, and we can make sucrose. We can send the sucrose out to the roots. We can feed the roots, right? We can do all that kind of stuff. So it depends on, does this cell need energy or do other cells need energy? Yeah. Okay. So now we think we're done, but we're not really done because we only really got 1 molecule out of the Calvin-Benson cycle. The other 5 need to go back in. But those 5 are carbon 3 molecules, right? We need a carbon what? 5. We need a carbon 5. So we're going to go from having 5 3-carbon molecules to having 3 5-carbon molecules. Right? It's too many numbers in carbon. I know. You do not need to know all the steps. All I really want you to know is that you're starting over here with dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. And what are you going to end with? Ribulose 5-phosphate. Now, is this the exact substrate? No, I need to get to ribulose 1,5-bisphosphate. So what am I going to have to do to get a bisphosphate? I'm going to have to phosphorylate it, so I'm going to have an investment of ATP. Okay, so now I'm going to undergo a little bit of a rearrangement, and then I'm going to phosphorylate my ribulose 5-phosphate to make ribulose 1,5-bisphosphate. And this is what we can use as a substrate in the subsequent Calvin cycles. Yes?
So on the last slide, you're always going to have the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate just because they resonate.
Yeah, they go back and forth.
You have to mention both.
Yep. Mm-hmm. Mm-hmm. But they're not that much different. It's just where the double bonded oxygen is. Is it on the end or is it in the middle? It just has resonance. It's fine. Okay. So how's that for the Calvin-Benson cycle? All right. There's a really nice animation that your book has that you should absolutely watch. Do y'all watch the animations? Yeah, they're really good. They're really good. Okay, that's it. I'm not talking about regulation. I'm not talking about anything else. I just wanted y'all to have an overview of how we really get energy in our environments, right? We get to eat all these plants. So go have a salad tonight. Yes, ma'am.
So this is when we like kind of introduce the pentose phosphate pathway.
Yeah, we're going to talk more detail about pentose phosphate. We will.
But this is one of the
But this is one of the ways that you could introduce it. But we're going to talk about it not the next chapter, but the following, which is carbohydrate metabolism. We'll talk about it there.
Photosynthesis is just, the carbon cycle is just one way to enter it.
Yes. Yes. Just what sections I covered. Just what sections I covered. Okay. I really think metabolism of the human body is more important for where y'all are going and what y'all are doing. But it's just nice to have an overview of how all this stuff works, right? Questions? Good. All right. So we're going to start 13 today because we have today for 13. Then we have next time a full class period. And then the following class period is going to be split half and half, right? So I'll teach for 45 minutes and then the other 45 minutes y'all will get to present. Okay, good. All right. And if you're presenting next week and have questions, always come and ask. Yeah, this is case study 1. What's that? Can't get anything right now? Yeah, try a different one. They don't work. Awesome. So we talked about making carbohydrates, but not inside of animals and not how we use those carbohydrates once we make them, really. So we'll talk about that. And the next chapter, we'll talk about real metabolism of all those carbohydrates. So carbs are the most abundant biomolecules. They absolutely are. You're going to be really surprised at how often you see them. And you know, we say, here's a protein, but that protein is not pure protein. Most proteins have carbohydrates attached to them. Lipids have carbohydrates attached to them. Lots of things have carbohydrates attached to them. So they get to be really, really important. Carbs are our carbon, hydrogen, and oxygen. That's all you're going to have on a carbohydrate, right? So you can start with that empirical formula, and then depending on how long the carbohydrate is it a simple sugar? Is it a complex sugar? It's going to get longer and longer. So you're going to hear them called glycans. Glycans, carbs, sugars, all the same thing. Don't get thrown off by the terminology. You probably just never heard it called a glycan before, right? So they'll call it a glyco group. So we have basically 3 types. Simple sugars. Give me an example of a simple sugar. Glucose, right? So anything that is a monomeric or dimeric really is a simple sugar, 1 or 2. Or really oligo, so get up to about 9. Then you get to polysaccharides. What's our most important polysaccharide in animals? Where do we store energy? In glycogen, right? Glycan, glycogen, that's us. Then you have what are called glycoconjugates. Glycoconjugates are like I was talking about earlier, where you have, for example, a protein with sugar attached to it. You have a lipid with sugar attached to it, right? So you've conjugated a sugar to something. So here are our examples, right? Simple sugars. You can have mono, di, and oligo. We said glucose is a good example, galactose, mannose, there's all kinds. Disaccharides. We've talked about sucrose, right? In the last class, we talked about lactose. Last time, not really maltose, I don't think. Did we? Maybe. Oligosaccharides. So oligosaccharides are in milk. We'll kind of talk about that. It's really cool. Raphanose, we'll talk about a bit. Then you start building them longer and longer, we get to polysaccharides. Polysaccharides can be insanely long. You can have some that are the same thing over and over and over and over and over again. Those are homopolymers. Glycogen is a homopolymer. It is the same unit repeated over again, right? Starch is another example. Cellulose is another example. Or you can have what are called heteropolymers. Heteropolymers depend on, you designate their description. These are disaccharide heteropolymers because you'll have two different monosaccharides that get repeated over and over again. So that's a disaccharide heteropolymer. It's 1, 2, 1, 2, 1, 2, right? These are really important for organization. In insect exoskeletons, the chitin in there, but also in things like your cornea, in your eye, you have keratin sulfate. That's really, really, really important. And I'll show you some examples of that kind of stuff. And then this is the glycoconjugates. So you name glycoconjugates with the last part of it indicating what predominates in the molecule. So a glycoprotein, what predominates? The sugar or the protein? Protein. Proteoglycans also have proteins and sugars, but what's different between a proteoglycan and a glycoprotein? Glycoprotein is mostly protein, proteoglycan is mostly sugar. So that's what you're seeing here. This is mostly protein with some sugar attached. This is mostly sugar attached to a protein, right? Then you can have glycolipids. Glycolipids, we don't really talk about the quantity. We just have one general classification where we say it's a lipid and a sugar. So the one that you probably heard about but didn't really connect as a glycolipid are our blood antigens, right? Our ABO blood groups. Did we talk about that one? A little bit? Yeah, a little bit. Okay. So here are a few of the individual monomers of sugar residues that are attached to either proteins or lipids or whatever it is. But these are our glycan groups, right? Our glycan groups. I don't really call them sugars anymore. We're going to start attaching them to things. Now, you could spell out the entire word, which would be really long, right? You could do the 3-letter abbreviation, which is sometimes nice, or your book is really nice in the whole book from here on out. When they have, for example, N-acetylgalactosamine, this is the symbol it's going to use, right? So do you have to memorize them? No. But if you're ever lost and you're like, why is it a green circle? You could come back and go, oh, that's mannose.
Should we know like the abbreviation?
Like glucose, galactose, mannose. And the other two big ones, I guess, really are the N-acetylglucosamine and the N-acetylgalactosamine. Those are your big ones. If you know the first like 5 for the 3-letter abbreviation, you're not really going to be lost, right? Those are the big ones. All right. So when we talk about sugars, we call it glycobiology, right? So in order to understand how sugars are used, right, you have to understand what happens to them in the cells. So Glycan Biochemistry, really what we can do is we can isolate, let's say, a surface glycoprotein, right? And maybe this is a surface glycoprotein that a virus uses to attach to a cell. Well, what on this glycoprotein is actually recognized by the virus? You can actually cut it out. You can do things like liquid chromatography, mass spec, and you can actually figure out exactly what the pattern of glucose molecules is, so you know what the virus is binding to, right? And then maybe you can design some kind of inhibitor. So understanding the structure and the sequence of those glucose molecules is really, really important. And that's really Glycan Biochemistry, right? And you can also understand, okay, which monosaccharides are coming in to make those, and how are they getting the individual monosaccharides, right? Because they can come from different sources. Glycan biosynthesis, that's where we're saying, okay, which ones come in and where and how are they assembled, right? So usually that's the ER, the Golgi, where we're putting them together, we're glycosylating our proteins, our lipids, or whatever it is. When we make the glycans, there are enzymes that facilitate those reactions, but there are so many different enzymes that catalyze those reactions that each enzyme is super insanely specific. Like if I want to add a glucose directly to a protein, well, there's a particular enzyme that's going to do that. Rather than adding a glucose to a galactose, I would need a different enzyme to do that. So the enzymes and their activities are very, very, very detailed, very specific. So then you have glycan diversity, right? You're going to have all these different glycan groups that are attached to different proteins, different lipids, right? And you can have classes where they do particular functions depending on the sequence of glycans that are attached to them. And then glycan recognition. So this kind of links into glycan biochemistry, where you can design things to disrupt an interaction between two cells, or an interaction between a cell and a virus or a bacteria or something like that. And I'll show you some examples later on down the road. But it's kind of amazing how diverse sugars are inside of the cell. It's a big I think area that wasn't studied until fairly recently. So here's an example of a glycosyltransferase. So this is an enzyme that is going to add a particular group to, and for example, this, what is this? What does SER stand for? Serine. Serine has what kind of a side chain? It has an OH, so it's a polar side chain, right? So when you think about linking sugars, what typically links, look down here, we'll kind of jump ahead. What links these two sugars? An O-glycosidic bond. So you really need an oxygen in order to link a sugar. So serine is a great place where you can do that on a protein because you have that oxygen there. So basically what you have is the sugar that is linked to UDP. What is UDP? Looks a lot like ADP, right? But instead of being adenine, it's uracil, right? So it's actually attached to a dinucleotide. The enzyme will transfer the sugar from the dinucleotide onto the protein. So now you have an O-linked glycoprotein. But usually it's not just one sugar residue or sugar moiety. Sometimes you'll hear it called a moiety. That's attached to a protein. Typically it's a really long chain. So now I need a different transferase. And I'm going to take my new sugar molecule. In this case, this is going to be GAL is galactose, right? This one was N-acetyl galactosamine. So here we're going to add our galactose, and it's the same thing. It's also attached to a dinucleotide. And now we can actually have that normal glycosidic bond formation occurring. And remember how we name those glycosidic bonds in biology 1, biochemistry 1? No, not really. We're actually not going to go in and have to rename them again. You're not going to have to. But do you remember what that refers to? Like if you have galactose beta 1,3, what's the beta? Up or down of the glycosidic bond to, what are you comparing it to? Like, how do you know if it's up or down? The carbon with the highest number that's outside of the ring, right? So you only label those as alpha and beta, right? If what? If the anomeric carbon participates in the glycosidic bond. Remember that? Yeah, y'all like sort of partially. And then the numbers just tell you which carbons are participating in the glycosidic bond. Yeah. They can attach to anything with an oxygen, but serine tends to be the most popular, right? It's the same thing with like phosphorylation, right? Depends on the enzyme that's actually doing the transfer, what it's designed for. So the enzyme specific not only for the sugar residue, but it's also specific to the amino acid residue. Yeah. Okay, so this is more like what you would actually see attached to a protein. See how we have a different residue, right? So you can attach it to anything that will allow that interaction to occur, and it can be an oxygen, and it can also be a nitrogen. So when we look at this is a much more typical length of a glycan group, right? If you have one, it's not typically a glycan group. Typically your group is a collection, and they can actually branch, and they do it really often. So you could draw it out, you could give the 3-letter abbreviations, or you could just do the picture diagram, which I really love. All right, so glucose and glycosidic bond refresher, right? Because I kind of figured. So if you're going to look at an individual monomer, right? What can we say? Which one is the anomeric carbon? How do I identify the anomeric carbon here? Attached to 2 oxygens. So carbon 1 here, it becomes carbon 1 because it's attached to 2 oxygens. What if you had a sugar that had this on it? This carbon becomes carbon 1, this carbon becomes carbon 2, and so on and so on and so on, right? What's that? But this carbon, see it? This carbon is still the anomeric carbon. Correct. Correct. Just be careful with that number. That always trips people up. It just always does. So I look at my hydroxide that's on the anomeric carbon. I compare it to my greatest numbered carbon outside of the ring. This one's going up, this one's going down. So it is alpha, right? And I would have to tell you if it was D or L, but what does the body prefer? Well, what does the body can only use is D. All the enzymes are designed for D. Remember when we drew them in the linear form and we looked at that last hydroxide farthest away from the what will become the anomeric carbon? And we said, is the hydroxide on the left or the right? If it was on the left, it was L. If it was on the right, it was D. I think I just did that pointed wrong. But my left and your left are different. Okay. Yes.
What was the other thing with linkage? Like if it's 1,4, it'll or 1,6.
Yeah, there are different linkages. So you can have the same monomer, glucose, right? And if you link it in a different way, it has a different bond strength. And we're actually going to talk about some of this. And so when you go from that linear form to the cyclic form, you can get a combination of alpha and beta, but one of them tends to predominate. So this is glucopyranose, right? It's the same thing as glucose in the cyclic forms. You can have that cyclicization occur where that hydroxide is sometimes below the ring or the hydroxide is above the ring. In the body, the beta tends to predominate by a little bit, not that much, but just a little bit. It's not bad. Okay, oligosaccharides. Oligosaccharides are really pretty interesting. So, you know, when you eat a lot of beans, what happens? You get gassy, you get really gassy, right? Why do you get really gassy? You have consumed an oligosaccharide that your body cannot metabolize. So it goes through your intestines until it gets to the bacteria inside of your intestines. The bacteria inside of your intestines are like, dinner, yeah. And so it breaks down the oligosaccharides that you can't, but it does it through fermentation. And when it does it through fermentation, guess what one of the products is? So you know, there's on the market, there's Beano, right? Have you heard of Beano? So all Beano does is it is an enzyme that will break down the oligosaccharides that you can't break down. So that by the time your food gets to the bacteria, it's already been broken down and you don't feed the bacteria, and the bacteria don't produce gas, and therefore you don't have gas, right?
Would that have a negative effect on your gut microbiome if you took that enzyme?
Well, if you change your diet dramatically, yes, like your gut biome changes depending on your diet, absolutely. So if you're used to eating beans once a week, right, it's fine. You probably don't have that much gas, right? But if you never eat beans, like you're one of those people who just didn't eat beans. I don't know how you live in South Louisiana and not eat beans, but, and then you had a bowl full of beans. Your gut is not designed for that kind of diet, right? And then you're gonna have massive gas. So your gut biome adjusts depending on what your diet is. So a lot of people who like go on a diet, right? Usually they shift to eating more of those fibrous, you know, any food with high in fiber. And a lot of times their gut biome is not adjusted to the high fiber diet. And so one of the side effects of then going on a higher fiber diet is, guess what? Gas. But then your gut biome will adjust. As long as you continue on the diet, your gut biome will adjust. Yeah, it's really cool. It's really cool. Okay, so that oligosaccharide that you can't digest in beans is called stachyose. So that is the enzyme that is the, I'm sure, sorry, the oligosaccharide that you cannot digest, right? Raphanose is another example of an oligosaccharide. The two that I'm showing you here are actually from milk. These lacto-in tetrose and lacto-in fructopentose. These two are actually found in mother's milk. So if you actually look at mother's milk, you have free monosaccharides. You have some proteins in there, and then you have some of these oligosaccharides. And these oligosaccharides are really actually important because, and I'm going to show you, I think I'll show you on a slide. Maybe I don't show you. I think I don't show you. So what these monosaccharides, these oligosaccharides can do is, it's really cool because they help to prevent bacteria from growing inside the baby's digestive system that aren't supposed to. So it actually, these oligosaccharides actually bind to the surface molecules on bacteria that are not supposed to be in the gut. So they act as like a sponge, and they suck up the binding sites on these detrimental bacteria. And therefore it's a protective mechanism inside of mother's milk to prevent uninvited bacteria from growing in the gut, right? Some bacteria is good. You have to have some bacteria in your gut or it's not working, right? But you can have pathogenic bacteria in the gut, which can cause serious problems. So these will bind to those bacteria that are not supposed to be in the gut. It's really cool. That, like, that's one of the things that mother's milk helps to do. Sure. Sure. Absolutely. Absolutely. So Raphanose is another example of an oligosaccharide. So this is what I was talking about. So it's not just beans. I know I said beans, but it's not just beans that can give you gas, right? Any of the ones that are actually based on raffinose, we can't cut. So raffinose is galactose, glucose, and fructose. So we can cut between galactose and fructose if they're separated, right? So if we have sucrose, our body can metabolize sucrose. But when you start adding galactose to it, we can no longer metabolize it. So what the enzyme in Beano will come and do is to cleave those galactose residues off. So now you don't have oligosaccharides that are going through your digestive system that the bacteria then love. Right. structural carbohydrates, right? Everything we've talked about so far, right, are going through metabolism. But plants use carbohydrates as structure. So cellulose, right, is the most common carbohydrate in plants because plants and fungi, it makes up the cell wall of fungi too. These are homopolymers. Homopolymers mean that it's the same repeating unit over and over again. So when you look at these are two, I guess I didn't label them, glucose, GLC, glucose. So these are two glucose molecules that are linked together in a beta-1,4 linkage. So it's the actual linkage between the two that are going to provide that rigidity. But here's the cool thing, right? You don't, in a plant cell wall, you don't just have one simple strand. You actually have another strand underneath and another strand and so on and so on and so on. And so all of those sugars, what do they have hanging out? What do they have? They have lots of OHs. Well, if you have hydroxyls, what's going to happen? What are you going to create? Not glycosidic, hydrogen bonds. It's the hydrogen bonds between the strands that add to that rigidity, right? So the glycosidic bond is really important, but then also important are the hydrogen bonds that link it, right? You can also have inside of cell walls something very similar to cellulose called hemicellulose, and it's basically the same thing except it branches. And then you can have pectin. So I want to show you inside the cell. Oh, was that in the last one? Oh, they don't show it. So in your textbook, there's a really nice picture. And in the picture, it's like a square cutout of the cell. And it's of the cell wall. And you can see like there are some really long tubes, and these are the cellulose, right? And then you have some thinner ones that are hemicellulose, because the hemicellulose can't have as many connections in hydrogen bonding because of the branching, right? So you got to think about, like, you know, it's like spaghetti strands in cellulose, but in hemicellulose you're branching, so the strands can't get as close, so you don't have quite as many interactions. But your book has a nice picture of that. Okay, chitin is another structural carbohydrate. So in this one, we have, in addition to having our glycosidic bond here, we have an additional chain coming off. So what is GLC-NAC? What is that abbreviation? N-acetyl, that's what the N-acetyl, you just start in the middle, N-acetyl GLC glucosamine. So you're going to have this repeating unit of these two N-acetylglucosamines, and then they're going to link together, right? So this is the repeating unit, and that's why they put the little N there. So this is in all the exoskeletons of crustaceans and then insects and that kind of thing, and also inside of fungi. So, but it just kind of depends on its orientation, whether you're going to have something really, really hard and tough versus not so much. Okay, so remember how I said that there were some of these glycans that were really important for arrangement of things inside the cell. So if you go back, like in the very beginning, you have, these are glucosaminoglycans. So these are repeating units, just like we saw before. But what you're going to see is they have typically these charged groups on them. Keratin sulfate, this one. Keratin sulfate, this is one, whoa, that you're going to actually see in the cornea. So in the cornea, you have proteins. Like, we know the cornea is completely clear, right? If your cornea were not completely clear, it's the outer layer of your eye, you wouldn't be able to see anything. So the arrangement of proteins within the cornea of the eye is insanely important. If you have a mutation in the enzyme that will generate keratin sulfate. And a lot of times what happens in the mutation is they lose the charged portion of the molecule. If that happens, basically at a very, very young age, you get a haziness inside of the cornea, right? And so it's very, very difficult to see through. Everything looks kind of fuzzy, right? So all of these are basically organizational glycosaminos, right? So arrangement of proteins within a cell and that kind of thing. Heparin, you know what heparin is? Yeah, what's heparin? It's an anticoagulant. I love that you did not say blood thinner because that is one of my pet peeves, right? So, you know, like aspirin commercials and things like that, they're like, oh, it's a blood thinner. Your blood does not get thin. What you do is you prevent coagulation from occurring. And that prevention is due to your charged group here. So if you lose this, you can no longer prevent coagulation. Very nice, thank you. All right. So now we're gonna switch a little bit, and we're gonna talk specifically about some storage sugars, right? So starch and glycogen. Starch is in what? Right, this is storage. Storage sugars. Starch is in where? Plants or animals? Plants. Starch is in plants. We have glycogen, right? They're very, very similar. But starch, you do need to know these. Starch has amylose and amylopectin and a few other polysaccharides, but the two big ones are amylose and amylopectin. You need to know those. Glycogen, on the other hand, is in animals, but it's the same thing. We use it to store glucose for energy. The deal with glycogen versus amylose and amylopectin is it has more branch points, right? More. And the question is, why are branch points important? So you got to kind of look at the structure and see. So let's look down at the bottom at C first. So if I want to store energy, I'm going to take my glucose monomers and I'm going to link them together. But do you remember we talked about a reducing end and a non-reducing end in Biochem 1? So if you have a non-reducing end, what does that tell you about the sugar? The anomeric carbon is participating in a glycosidic bond. If you have a reducing end, the anomeric carbon is free, right? So write that down. Reducing tells you your anomeric carbon is free. Non-reducing anomeric is in glycosidic bond. Here's the deal. The enzymes, right? If I say a bear walks in the classroom, I need to run out the other door, right? I need a sugar rush to go through my body in order to run as fast as possible. Y'all are gonna beat me. I'm gonna get eaten by the bear, but it's okay. But everybody's gonna want that sugar rush, right? So we didn't just eat anything. So where do we get that sugar? From our stores. But here's the deal. You can only cut off individual glucose monomers that have a free anomeric carbon, right? So basically you can only cut off the ends. Everything in the middle is all participating, right? They're all participating in glycosidic bonds. So you can only cleave them if you have a reducing end. So if you look at things like starch and things like glycogen, have y'all ever, this is gonna date me. Have you ever used or played with a Kush ball? You know what a Kush ball is? Okay, so it's this rubber thing and it has these like finger-like projections. Y'all know what I'm talking about? And it's like rubbery, right? Okay, so when you compare the two, I'm gonna come back, yes, like that, that's a Kush ball. Yes, okay, that. So when you talk about, I don't know. The cool ball, it's a Kush ball. It's a Kush ball, that's what it is, it's a Kush ball. So if you think of storage sugars as a Kush ball, right? These individual fingers are the only places where you can actually take an individual glucose, right? So the more fingers you have, the quicker you can put forth a stream of sugar into the bloodstream, right? So think about a plant. Is a plant going to really need a huge excess of sugar really quickly? Are they running anywhere? They're not, they're not running. But how about us? We hope not. It'd be interesting. Yeah. But what about us? Yes, we are going to need a much higher increase. So if you look at the storage forms, right? If this is our plant, right? It has so many fingers. Well, if I look at the human version, what's actually going to end up happening is we're going to have more branch points. So we're going to have more of these finger-like projections that are going to come off of the main chains, which means I now have more ends that are available to release sugar into the bloodstream. That's right. You have mini Kush balls inside of you. Microscopic Kush balls filling your body. Yes. What's that? Yeah. Okay. So that's amylopectin and glycogen, right? So if we compare the two, kind of make a comparison. How much time we got? We still got 20 minutes. Okay. So if we compare the two, so I'm comparing everything in C, amylopectin and glycan. And glycogen. Why did I say glycan? I know, it really is. Okay. So if I look, and then I also want to talk about amylose, right? So amylopectin and amylose both make up what? Starch, which is plant storage. Right, plant storage. So if I compare the two, and I look at amylose is actually going to be linear. So the picture up above here is amylose. When I say linear, have you seen, if you come to my office, you can see this. On an old phone, you know the cord that does the helix thing? Okay. Yeah, no, y'all don't have any of those at home anymore, right? The cords on the landlines, exactly. So that's the spiral that amylose has. It is linear. It does not branch, but it does spiral. Okay, so we call it linear, but it has a helix, a helical shape, right? Okay, so both amylopectin and glycogen, on the other hand, are branched. And then the other things that's different are the type of glycosidic bond we have. In amylose, what we have is an alpha 1,4 linkage. That's what gives it that helical structure. Amylopectin, on the other hand, is still an alpha, but it's an alpha 1,6. And it doesn't branch very often. It only branches every like 15 to 30 residues or glycogen or glucose moieties. You have a branch point. Whereas glycogen, even though it has the same linkage as amylopectin, it branches more often. So it branches every like 8 to 12 glucose units. So glycogen is going to be able to give us that quicker increase in blood sugar than amylopectin, which is really what we need. So the two that look like Kush balls are the branched ones. Then the one that looks like a phone cord is the amylose. Yeah. And the reason they look different is because of their linkages. Amylopectin is the Kush ball. So this is a Kush, K-O-O-S-H, Kush, I think, with few fingers, right? I'll call them fingers. And this is, and then glycogen is Kush with many fingers. Just to give you a visualization. And then the amylose is the phone cord. Things you never thought you'd talk about in biochemistry, right? All good. About a Kush ball, right? And it's the same thing. Go ahead.
What did you say about the body can't, if your body can't digest, it says beta?
Beta. So look at like chitin, beta 1,4. We can't digest that. That is roughage, is what that is.
Like when you said the beta D. Beta D glucose.
So you're talking. Whoa.
No, the beta D glucose.
Here. So this one, yeah, we can't digest the beta 1,4, right? Because you can eat when you eat plants have both starch and they have cellulose, right? You can digest the starch, but you can't digest the cellulose, even though that's the majority of the sugar inside of a plant cell is the cellulose. We can't digest it. So that's the roughage, right? That's why when you eat like things like celery, right? You're really not getting much out of it. You're just enjoying the chew. Okay. I did this one. Okay. Good. Oh, the other thing I wanted to say before we went on, right, is we talked about you could only take away, right, when you're on a reducing end. You can only add to a reducing end too, right? If you're building glycogen, you can only add to a free anomeric carbon. And you can only remove if you have a free anomeric carbon, because you're going to have something to attach it to that UDP molecule. So that's going to be really, really important. I'm sorry, go ahead. So the anomeric carbon, you need that free anomeric carbon. That's the reducing end. So either you need it in order to bond to the chain, to continue the chain, right? You're adding it to the glycogen that's already there, or you need that free anomeric carbon to associate with the enzyme that is going to go in and cut it. We haven't talked about it yet.
Oh, okay.
That's a transferase, but we haven't talked about the enzyme that cuts it.
I thought I heard UDP, so I was like,
Well, yeah, and then you're going to have to hook it to a UDP,
oh, okay.
right? Because that's how the enzymes that do that transfer, the sugar moiety is attached to a UDP, then we move it. So it's always, whenever you're taking away sugars and adding sugars, they have to be attached to something. So you have to have that anomeric carbon available for that interaction to occur. All right, I think I'm going to stop there, because we'll have 2 and 3, and then I have a little activity next time that we'll do. So that'll work out perfectly. So I'll get a few minutes. How about that? We're done for today. Calling it for today. How about that? Yeah. Okay. Y'all have a good weekend. Let me end this.